This is the first of 3 videos in which we're going to look at cell physiology, and I just want to remind you that we'll be looking at both structure and function of the cell. Before we get started, I just want to remind those of you who might be taking class with me, it's important that you take notes of these. I prefer that you do handwritten notes. It's also important that you write down questions, and I want you to have those ready at the next class because I'm probably going to randomly call on people to make sure that they do indeed have questions, and you should have those ready to hand in at the end of class because I may collect them. All right, in the first video we're going to look at an overview of cell structure, what it's made up of, and then we're going to go ahead and we're going to focus some on the structure of the cell membrane. And here, in fact, you can see a great picture of a cell membrane. And so we'll talk in detail about what it is that you see there. Now, let's start to talk about cell. The word cell comes from cellula, and it is Latin for small room. Now, the reason that that is the case is because with the first microscopes, one of the things that they looked at were thin slices of cork. And in cork, what we just have are the empty walls of the cell. There's nothing in there, and so it did look a little bit like a room to the people who were looking at it. That's where the word cell comes from. The cell is the structural and the functional unit of all living organisms. Well, what does that mean? It means that it's the smallest unit that can perform all vital physiological functions. You only need one cell, but you got to have at least one complete cell. Each cell maintains homeostasis at the cellular level. And this is important because homeostasis at tissue, organ system, and the organism level is due to the coordinated action of many, many cells. In fact, about 37 trillion of those guys. All right. When you looked at the video on the scientific method, we talked a little bit about theory, what theories are, and we even looked at a couple of different theories, including the cell theory. Well, now we're going to go over that again, talking about cell physiology, so this becomes important. The cell theory suggests that all life forms are made from one or more cells. It also says that cells only arise from pre-existing cells via cell division or mitosis. And here you can kind of see a little bit of an illustration of that. Here's our original cell. It goes through mitosis, and we have 2 daughter cells, but we don't stop there. The daughter cells will divide, those cells will divide, and so on and so on. And so we're constantly replacing cells. Even neurons, which we used to say, well, once you got to be an adult, you didn't create any more neurons. Turns out, well, in fact, you can. Here we can actually see some photomicrographs. Here it is cell division, which is almost complete. You can see 2 complete nuclei, and the phospholipid bilayer membrane has almost been completely broken apart. And so here we can see that the cell is in the final stages of cytokinesis. Here we can see that this particular cell is in the earlier stages of mitosis. The 2 nuclei aren't quite completely divided yet. And here is one that's in the very, very early stages of mitosis. The cell is also the smallest form of life, and again, it is the structural and functional unit of life. Cells contain the hereditary information. That is the DNA, which we find inside the nucleus. And through division of the nucleus, that hereditary information is passed on to all the daughter cells. It goes from cell to cell to cell. All cells are the same in chemical composition. Now, that doesn't mean that one particular cell doesn't have more of a chemical than another cell. And so, for instance, myocytes, lots and lots and lots of protein. So far more protein in myocytes than there is either carbohydrate or lipid. On the other hand, adipocytes, lots and lots of lipid, not so much protein and not so much carbohydrate. Now, there aren't any cells that I can think of which are mostly carbohydrate, but we can think of a cell like a hepatocyte, a liver cell, and it tends to be higher in carbohydrate than other cells because it stores carbohydrate in the form of glycogen. And here you can see the generalized structure of a cell, and this shows components that we find in all cells, but not to the same extent. The energy flow of life occurs within cells. Later in the semester, we're going to go through cellular respiration, go through energy metabolism, and we'll talk about it more in terms of the whole body, but it happens within cells. Well, how large is a cell? What I'd like you to do is pause the video and just think about that for a minute. And so I hope you realize that cells are microscopic. That means they're so small you can't see them with the naked eye, with the exception of one cell. And so the human egg can, under the right conditions, be seen without a microscope. But most require a microscope to be seen. Here we're going to look at a quick little video that gives us some perspective on this. And so here we can see, look, it's a little bit large, and this is a coffee bean. Lots of you have seen coffee beans. This is, again, slightly larger than a typical coffee bean. Here's a grain of rice. Here's a sesame seed. We'll blow this up a little bit. Look, here's a grain of salt. If you have some salt, you might pour a little bit out on your desk and spread it out and look at it under a strong light and see if you can find a single crystal or a single grain. I'll blow things up a little bit. Oh, look, here are some organisms that probably you've studied in biology, either in high school or perhaps here at DePauw. And so here is an amoeba, and here is a paramecium. Now we can finally see, oh, there's that human egg cell. And so, oh, look, 130 micrometers. You can also see down here a skin cell. A skin cell is also a type of epithelial cell. Now we can also see a sperm, and you can see it next to the egg. Here's a photoreceptor, which we find in the retina of the eye. And here is a red blood cell. And so, again, pretty small. And you have trillions of those guys. Here we blow things up even more. There's a little bit of baker's yeast. And if you're a vegan, it's good to get some baker's yeast because that helps to ensure that you get the vitamin B12 that you need. But here are some things that make up cells. Here's a chromosome, and it's in the highly condensed form. Many of you may recognize that. Here are a couple of organelles, a mitochondrion and a lysosome. And then there's a bacterium, which could make you sick. The truth is, you actually have more bacteria on your skin and in your gut than you have cells in your body. So more than 40 trillion bacteria. All right. Now, look, you can start to see some viruses. There's a measles virus, which is a pretty big one. And here is the HIV virus. Now, I wasn't really going to spend any time with viruses, but since we're in this pandemic and we're all concerned about the coronavirus is just slightly smaller than an HIV virus. So it's about 125 nanometers. Now, the nice thing is that HEPA filters can take out particles as small as 10 nanometers. And so a HEPA filter can actually filter out a lot of different viruses. And then we go down a little bit more. Here again is an organelle, one of the really tiny ones. Here's a ribosome. And then we can see some molecules. And so antibody is protein, hemoglobin is also protein. tRNA is a nucleic acid. And then we'll blow it up just a little bit more, and we'll look at, here's a phospholipid, which we're going to talk about a little bit later on. Here's a glucose molecule. So that gives you some perspective on the size of a cell. Whoops, not what I wanted to do here. Sorry, I'm not sure what happened. All right, so now we're back to this. Here, this is a table which lists certain cells, and it gives you the volume of those. And so you can see, again, sperm cell, pretty small when we look at the volume of it. The egg cell is the biggest. A fat cell is pretty big, but still nowhere near as big as an egg cell. I obviously don't expect you to memorize this. It's just there to give you a little bit of perspective. All right, well, what do cells look like? And again, what you might do is pause, give this a little bit of thought. Have I seen a cell? What did that cell look like? Well, this shows you some of the diversity of different cells. Oftentimes we think of a cell as kind of a spherical blob. Now, what you need to keep in mind is cells are three-dimensional, but all of these pictures are two-dimensional, so you need to think about it. Well, we just saw an egg cell and we just saw a sperm cell. An egg cell looks a little bit like, oh, a typical cell. A sperm cell, nothing like that. Here is an adipocyte. So look at all the fat that's in there. Hardly any protein, hardly any carbohydrate. This is a smooth muscle cell. And so you can see it's a little bit cigar-shaped. Here are some red blood cells. The red blood cells are actually shaped a bit like a lifesaver. Here are some white blood cells. This is a bone cell. And here is a neuron. Look at that thing. That's crazy. So there you go. And then here are some columnar epithelial cells. So not only great diversity in the size of cells, but great diversity in the shapes of these things as well. Again, how many cells do you have? Well, quite a few. And so the latest estimate that we have puts it at about 37 trillion cells, or just under 40 trillion. So if you remember this, then good, way to go. Now, again, this is our best estimate. So as we get better at studying things and as we improve our technology, we're probably going to refine this estimate. Well, how many different types of cells are there? Those 37 trillion cells represent about 250 different types of cell. When I say type, well, here we can see several different types of cells. We could break this down even more and look at red blood cells. We could look at lymphocytes. We can look at macrophages, and so lots and lots of different things. All right. By the time we get to the end of Anatomy and Physiology 2, you might not know 250 different types of cells, but you should know 20 or 30 or perhaps even more. So again, here is kind of our generalized picture of a cell. This isn't any particular cell. It's not a myocyte. It's not an adipocyte. It's not an egg cell. It's just a generalized cell, which shows you all the different parts that we might find in a cell. And again, some of you who've had some biology and whatnot, or a good biology high school class, you probably can label most of what you see there. And if you can't, you need to be able to by the time we're done with this section. And so there we go. There are three principal parts that make up all cells. Okay? And the outermost part is the cell membrane. Okay, cell membrane, look at it, here it is. It's a phospholipid bilayer. Those of you who had some biology and have studied plant cells have cell walls. Human cells don't. So the cell membrane and the cell wall are different. They're not the same. Earlier in the semester, we talked about the serous membrane. It also is different than the cell membrane. The serous membrane is made up of epithelial cells. The cell membrane is made up of phospholipids and other things. The second part of the cell is the nucleus. And here you can see the nucleus. It's this whole piece. I don't know what color you'd call that. It's kind of a puce or pinkish purple. I don't know what it is, but it's the whole thing, not just that darker blob in the middle. This is the nucleolus, which is part of the nucleus. Now, the nucleus technically is not an organelle. Okay, here you can see what we've done is we've isolated the nucleus, and you can see that it actually has two membranes, an inner and an outer. There it is. And notice right here, the endoplasmic reticulum is a continuation of the outer membrane of the nucleus. So there we go. And then finally, we have the cytoplasm, along with the organelles and the inclusions. So those are all part of it. In this particular cell, the cytoplasm with the organelles and inclusions is this pinkish part. So from the outside of the nucleus to the inner layer of the membrane, cytoplasm. There we go, cytoplasm. Okay? All right, now we're going to go ahead and focus on the cell membrane for the rest of this video. And first we'll talk about the functions of a cell membrane. Well, one, the membrane forms a barrier between the extracellular fluid and the intracellular fluid. Those two fluids are different components in the extracellular environment than the intracellular environment. And so we have these phospholipids that create a hydrophobic barrier that keep those things separate. Secondly, the membrane determines the movement of substances into and out of the cell. And so we get exchange with the environment. And we say that the membrane is semi-permeable. It's not totally impermeable, but it's not permeable to everything. So it's semi-permeable. The membrane is also responsible for recognition of other cells and communication with other cells. And that's largely due to what's called the glycocalyx that's out here. And we'll look at that in a bit. And when we think about the structure of the cell, it's largely due to the cell membrane in association with the cytoskeleton. So the cytoskeleton is here, close to the inside layer of the membrane, helps to give it shape. Okay? Well, what is the membrane made up of? The membrane is made up largely of lipids, about 45 to 50%, a little bit, maybe a little bit less depending on what we're talking about. About the same protein. So again, some cell membranes a little bit more lipid, other cell membranes a little bit more protein. Now, when I learned about the structure of membranes, we said, oh, they're mostly lipid, and only a little bit protein. We've learned a lot since then. We also have carbohydrate as part of the cell membrane, but we only found it on the outer surface and not to a great extent. So there's not a whole lot of carbohydrate there. All right. Well, what are the types of lipids found in the cell membrane? The major one, the biggie, is phospholipids. So the cell membrane is a phospholipid bilayer. I hope you remember from when we did chemistry, we talked about the amphipathic nature of these guys. And so you should think about that and say, oh, do I remember the amphipathic nature? I'm going to go up here and point to this and say, oh, this should tell you about that. All right, another important lipid is cholesterol, and as I told you when we did chemistry, you don't need to memorize this. You don't need to be able to draw it, but you should kind of recognize it. If you see something that looks like this, you should be saying, I think that's cholesterol, or maybe it's something that's derived from cholesterol. And then we also have lipids which have carbohydrate moieties on them, and we call these guys glycolipids. Here's that zigzaggy carbon chain. And so again, at every angle, carbon, 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 so on and so forth. And when we say a carbon chain that's nice and long like that, and we're talking physiology, you say, pretty sure that that's a fatty acid. And here you can see a different representation, a few extra carbohydrate moieties on here. But again, both of these are examples of glycolipids, and I guess I should say all three of them are examples of glycolipids. All right, now, by the time we get done with this and you've studied this and practiced it, you should be able to draw a schematic diagram of a cell membrane and be very complete with it. Here is our phospholipid, which is amphipathic. So remember, phospholipid, like a triglyceride, has a glycerol backbone. There it is right there. And then it has that polar head because it's got that phosphate group in it, very polar. And so it's going to associate with water. Water's polar. Phosphate group is polar. Good deal. Way to go. Here are the fatty acid tails, which are non-polar. Don't want to associate with water. Notice here that we actually have one which is monounsaturated. Look very carefully and you can see there's a carbon-carbon double bond. Kind of hard to tell, but you look, you know it's a carbon-carbon double bond because we've taken away a hydrogen from here, taken away a hydrogen from here. So it's got to be a carbon-carbon double bond. Okay. Here is a schematic representation of a phospholipid. Almost all physiology books show phospholipids this way. And so here is the red part that you see here is both the glycerol backbone and the phosphate head. And as we just said, hydrophilic. It will associate with water. Here are the nonpolar fatty acids, hydrophobic, don't want to associate with water. And so what does that mean when we throw a whole bunch of phospholipids into a watery solution? Oh, they're going to associate and they're going to put their polar heads out towards the water to the outside and in towards the water to the inside. And the fatty acid tails are here in the middle, and they make it yellow to give you the indication that that's kind of oily because it is lipid. It is lipid. And so now we can have a solution in here that's different than the solution out here, and it's protected. It's protected. Here you can see more detail about what a cell membrane looks like. It's a phospholipid bilayer, but it's got lots and lots of other stuff in there. Look at all the phospholipids. And again, we can see the glycerol and phosphate head and the fatty acid tails. What you see in blue are the proteins. And so you can see a number of different proteins. We're going to talk about protein channels and things like that in just a bit. But you can see that in a couple of cases, the protein goes all the way from the inside to the outside. All the way from the inside to the outside. And look, oh, those are called integral proteins. Integral proteins are going to pass completely through the membrane. In other cases, you can see that the protein might be on the inside and it projects into the membrane, but it doesn't go all the way through. Or the protein might be on the outside and it projects in, but it doesn't go all the way through. Now, those are what are referred to as peripheral proteins. And if I ask you to label a membrane or to draw one, you got to give me both. You got to tell me, oh, there are some peripheral proteins, oh, there are some integral proteins. Don't give me just one or the other. All right, now let's go ahead and talk about other lipids. Look, here's cholesterol. Cholesterol is periodically put in here. And again, some membranes a lot more cholesterol than others. The cholesterol makes the membrane just a little bit stiffer, not quite so fluid. And so it doesn't just float off and float apart. Here are our glycolipids. And so they've made the glycolipids kind of neon green. And you can see the lipid portion here and here, but not in those guys. And so these are the carbohydrate moieties, and there are the fatty acid tails. And then we can also see, some glycoproteins. And here are the carbohydrate moieties, and here's the protein part. Now, the proteins are just shown very, very schematically, as you can see. All right, I don't think I missed anything, but here's a different depiction. And so again, you could go ahead and look at this one and compare the two. I'll just remind you that the glycolipids and the glycoproteins make up part of what's known as the glycocalyx, which is out here. It associates with the extracellular matrix, and it's involved in cell-cell recognition and also cell-cell communication. All right. Then last thing we'll talk about is this cytoskeleton, which is not technically part of the membrane. Those are organelles that are part of the cell, but they're associated with the membrane. And again, they help to give certain cells shape, and they also are responsible for movement of some cells. And cells move around, especially cells like white blood cells. They move through the bloodstream, but they also can get out and get into connective tissue, move to different places. Cells are phagocytic. They can engulf other things and eat them up. And so it's the cytoskeleton that allows them to be able to do that. All right, well, role of proteins in cells. Well, to an extent, they help to provide some structure. They can also be enzymes. And so if you look at the mitochondria and the inner mitochondrial membrane, lots and lots of enzymes in that phospholipid bilayer. They serve as receptors. And so they could be receptors for neurotransmitters or hormones or other things. They also are transporters. Now, these transporters we're going to talk about from here on out. They become critically important in understanding membrane potentials and action potentials in the nervous system, but also important in digestion, the way that the kidney works, the lungs, and on and on and on and on. And we have some transporters that are just open channels. This is like a little piece of a straw. Here are the walls of the straw, and there's the middle part. And so here's potassium, and it's going to diffuse from high concentration in the cell to low concentration outside. This is just always open. It's a leak channel. And so not only do we have potassium leak channels, but we also have sodium leak channels. Sodium, on the other hand, it's in high concentration outside, low concentration inside, and so it diffuses into the cell. Here you can see that they're showing that there are more potassium leak channels in a neuron than there are sodium leak channels. We also have channels with gates that can be opened up by voltage. We use a battery to open these up. This is a critical one. This is the voltage-gated sodium channel, and it's got an activation gate, which is right through here. Here the activation gate is closed. Here it's open. Sodium goes in. And it also has an inactivation gate. Inactivation gate is open. It starts to close, but it just is very slow. A lot of sodium gets in before it finally closes. And then we have ligand-gated ion channels, and this actually shows you both things. It shows you the channel through the protein, but it also shows you these little receptor sites on it. And so the ligand in this case is acetylcholine, which is a really, really important neurotransmitter. And you can see that if ACH binds with both of the receptors on this ion channel, it opens up. And now sodium moves from out to in, goes down its concentration gradient, and potassium moves from in to out, goes down its concentration gradient. Well, once we get too much sodium in here and too much potassium out there, we've got to move them back to where they belong. Well, now we need to use these transporters known as pumps. Pumps are taking something from low concentration and moving it back to high concentration sodium from inside to outside. So again, don't forget, leak channels, sodium went in, the pump is going to move it back out. Potassium is going to go from outside to inside. It's also going down its concentration gradient. Well, that's work. It requires the input of energy. We need to break down ATP to make that happen, right? There are other pumps, but every book I've ever looked at, I look through books, I haven't done it for a little while, maybe I should go back. The only pump they ever show is the sodium-potassium pump. Well, here's another one, a calcium pump. And here it is in the membrane of the sarcoplasmic reticulum. This is endoplasmic reticulum of muscle. Here we have low calcium concentration. Here we have high calcium concentration. The calcium is being pumped to this side. Need to use ATP to make it happen. proteins can also be marker molecules, as can lipids. And so again, the glycoproteins and the glycolipids that are on the outside make up part of that glycocalyx. And so here we see O glycoprotein, O glycoproteins, okay, and also glycolipids. But again, carbohydrate moieties, one or more, attached to protein. Carbohydrate moieties attached to protein. And then proteins also join cells together. And so we talk about intercellular joining between cells. And usually it's parts of the cytoskeleton that will do that. And so you can see protein from this cell is moving across to this cell, and we make a nice junction right there. And there are all kinds of different junctions. We're not going to worry about those right now, but here you can see an interlocking junction known as a tight junction. Here's a different one known as a desmosome, and then here are gap junctions. So we'll talk more about some of those later, and we will talk about gap junctions a lot because they become very, very important. Well, this is just going to take a quick look at what proteins really look like. All of those pictures I just showed you, just schematics. That's not what proteins look like, and you know that because we talked about it. Here is a transporter known as a sodium glucose linked transporter, and it's used to move glucose across into the cell. Well, look, here are the amino acids that make up this guy. And so it's a protein. You can see it weaves its way back and forth through the phospholipid bilayer. That's what this gold or mustard color thing is supposed to represent. There are the phospholipid heads on one side. There are the phospholipid heads on the other side. Okay? And here's the schematic representation of that sodium glucose linked transporter. Sodium goes down its concentration gradient, and it drags the glucose with it. Here's another one that works on glucose can also move across a cell membrane via facilitated diffusion. That means it has to have some molecule that helps it move across. And what it uses is a protein called a glucose transporter, or GLUT molecule for short. Again, you can see there are the individual amino acids that make this thing up. And so let me see here. There's a GLUT molecule schematic representation. Here's what it really looks like. Here's our voltage-gated sodium channel. Not really sure why I stuck that in again for you, but I'll go through it real quickly. There's the activation gate. Right now it's shut. And here's the inactivation gate. It's open. So we use some voltage. This one will open up and sodium will go in. And then this one will close. Here's a different depiction of a voltage-gated sodium channel. So when we get to the nervous system and I say, hey, do you know what a voltage-gated sodium channel is? You all better say yes. I couldn't find a nice depiction of the protein structure of the voltage-gated sodium channel, but I found one for a voltage-gated calcium channel. So here is the voltage-gated calcium channel in the closed state. The calcium can't fit through there. Here it is in the open state. Now the calcium can go through. Oh, and look here. You should recognize these from our discussion of chemistry of biomolecules. Look, here is the alpha helix. There's the secondary structure of this piece of protein. Well, here it is all folded up. It's folded. There's the tertiary structure of this protein. Same thing for the purple one. And then you can see that there are 4 protein chains that are all folded up, that are associated with one another. There's the quaternary structure of this protein. And here's the calcium. It's going into the pore, can't go through because it's closed. This is looking at that thing from the top. We're looking down into it. There's the pore, and there's the calcium trying to pass through. Once the voltage opens it up, the calcium will indeed pass through. This is a receptor molecule. All right. I wasn't going to do this, but I'll go ahead and do it quickly. Again, pandemic, immune system. Okay. What we see here are proteins that are in the cell membrane that play a really, really critical role in the immune system. This is a major histocompatibility complex, and we've got 1 and 2. And number 1, this tags your cells as yourself. That tells your immune system, leave these cells alone, don't bother them. Number 2, when you have been infected by something, it tells it tells the immune cells that now they have to go into action and they have to deal with it. So that's what these MHC molecules are. They're part of the immune system, which we'll talk about a lot when we get to ANP2. All right, now we're going to finish up by talking about how the membrane is fluid. It's fluid. It can flow. It can change its shape. It can do things. Well, what does that mean? It has a lot of important consequences. And so one is that it can distribute molecules and they can move from place to place within the membrane. They can leave the membrane. They can enter the membrane. And here you can see that the membrane can also merge with other pieces of membrane. And so here is a vesicle. Remember, three-dimensional. It's really like a little bubble. It's not like a disc. And so the membrane of the vesicle comes up and starts to merge with the membrane of the cell. It merges completely and opens up. And now this protein that's in here is released to the outside. That could be hormone. It could be neurotransmitter. Okay. Another thing that we can see here is our rough endoplasmic reticulum. We've made some pre-protein and packaged it into a vesicle. Again, a little bubble, not a disc. That travels across to the Golgi apparatus where it fuses and gets released in here. The Golgi modifies it and changes it and then packages it and sends it out. It could be used in the cell. It could become part of the cell membrane. Remember, lots of protein in there. Or it could be released to the outside, as we just looked at there. Another important consequence of the fact that the membrane is fluid is that small damage can be repaired easily because the phospholipids just flow into the damaged area and fix it up. And everything is good. What we're looking at down here is part of what we call the endomembrane system of the cell. And so we've got a number of organelles that have phospholipid bilayer as part of them, and they can send vesicles back and forth to one another. And so that brings us to the end of video number 1. Video number 2, we're going to focus more on nucleus and a couple of the organelles.